and I'll turn it over to Katie. Thank you.
Okay, hi everyone. Thanks for that really nice introduction. I'm Katie Driscoll, and I'm just gonna jump in. So I have, a short presentation, but it's packed with a lot of information. So I wanna make sure to get through all of it. So I'm gonna be talking about some of the work that I'm doing in northern New Mexico at some stream restoration sites, and then cover two topics that Katelyn suggested to me when we were talking about me doing this presentation that might be pretty relevant to this group. One are before-after control impact or BACI study designs, and the second is discussing indicators for ecosystem function. So I'm currently involved in some restoration work in northern New Mexico at two different sites. One on the Santa Fe National Forest in the Jemez at San Antonio Creek, and the second in Vidal Creek in Valle Vidal up on the Carson National Forest. So these systems have been exposed to a history of multiple interacting stressors that include overgrazing, timber harvest, and mining. And so the results of those stressors has been down cutting and incisement that's disconnected the channel from the floodplain. So managers in these systems are using two different treatment types to address this degradation. In the Jemez, they're installing beaver dam analogs. And then up on the Carson, they're using plug and pond treatments. So I'm involved in this work because I'm studying the effectiveness of these different treatments for recovery of ecosystem function. And to do that, I'm using a before-after control impact study design. So kind of in general, I'm of the view that restoration projects are really field experiments, and that if monitoring is conducted within an appropriate study design, you can kind of come up with this win-win situation where managers meet their monitoring requirements, but we can also draw some, statistically robust conclusions regarding how effective different treatments are. So there's an ongoing debate about the most appropriate study designs for drawing inference from field studies that involve non-random assignment of unreplicated treatments like restoration. But in general, all of the, best approaches are often different takes on before-after control impact. So, how I have found success, in this collaboration in northern New Mexico, as well as a couple others, in communicating the benefits of doing a BACI study design, are starting with really early discussions with managers and stakeholders focused on what their goals for the project are and what their plans for monitoring are. And out of those conversations, it's usually discussed that they want to make sure that restoration is really effective, that they're applying effective treatments, but that there's also resource constraints, especially when it comes to monitoring. So from there, I think it's, when I have worked with managers to apply BACI, making sure that they're familiar with the pros and cons of the different monitoring study designs, and then especially providing examples of what we can do if we collect data within a BACI framework has really helped get these projects established on the ground. So, one really common approach for monitoring restoration are control impact designs, where you compare a control and impact site and can conclude that restoration has an impact if the sites differ from each other. But the flaw here is that there's no two sites that are identical. We expect them to vary even before restoration's applied. And so this approach can confound the effect of restoration with processes that produce spatial variation. And so the end result can be a high rate of type 1 error where we conclude there's an effect of restoration when there actually wasn't one. A second approach is doing before-after monitoring. So you compare the state of the impact site before and after restoration happens and conclude that there's an effective restoration if the periods significantly differ. But we also expect sites to vary naturally over time. And so this approach can confound restoration with processes that produce temporal variation. And again, have us accept that there was an impact when there wasn't actually. That being said, before-after designs can be a good alternative to BACI if you have a long and dense time series data set and well-behaved temporal dynamics. So then that gets us to the BACI framework, which is what I'm using in my research up in northern New Mexico. Like I mentioned, there's several different ways to do BACI. I'm using a paired series approach where I monitor both impact and control sites several times in the before and the after periods. But this approach attempts to deal with the temporal and spatial variation by monitoring control and impact sites both in the before and after periods, and ends up lowering that rate of type 1 error associated with the other monitoring approaches. So the traditional ways of analyzing BACI data are to start with this equation to determine the effect size. So you come up with, whatever your parameter of interest is, and determine the mean at the impact and the control, the difference between them in the after period, the difference between them in the before period, and subtract those from each other. And then to determine if the restoration has had a significant impact, you're really interested in this differential change over time between the two sites. So you use linear mixed effects models in ANOVA to study this period treatment BACI interaction term. And if it's significant, you can conclude restoration has had an impact. So, what that looks like with an example from one of my sites, this is stream temperature data from San Antonio Creek up in the Jemez. The blue line represents the impact site, and the orange represents the control site. So I have, time series data in this plot on the left, and this gray bar represents when the Beaver Dam analogs were installed in the impact site. So I calculated this effect size using that equation from the last slide, came up with 6.6 degrees Celsius. But then when we look at the period treatment interaction term that's in the bottom of this black box, the p-value here is 0.12. So we would conclude in this case that Beaver Dam analogs did not have a significant effect on stream temperature. Okay, so an alternative to that traditional way of measuring BACI data is this Bayesian approach that was covered in this paper by Mary Connor and others that came out in 2016. And unfortunately, I haven't had a chance to do this with my own data, so I have an example from this paper. But what I found is when I share this information with the managers I'm working with, this is the analysis that they're really excited about because the results tend to be pretty easy to interpret and more intuitive, especially for non-scientific audiences. So for this analysis, this is some example data that the authors of that paper collected in some streams in Oregon. So you have the black line is Bridge Creek, which was treated with Beaver Dam analogs. And the dashed line is Murderers Creek, which is untreated control sites. This plot is showing the before period and the after period with the vertical black line representing when Beaver Dam analogs were installed between 2009 and 2010. So in this case, they were measuring juvenile survival of steelhead. So they come up with the juvenile survival in these at both sites over time, and then calculate what they call an R ratio, which is just the survival at the treatment compared to the survival at the control. So this plot, that R ratio is represented by the light gray line. The dashed line right at 1 represents no difference between the treatment and control. And if the R ratio is plotting below that dashed line, it suggests that survival at the treatment is less than the control. If it plots above the dashed line, survival at the treatment is greater than the control. And then these dark black horizontal lines represent the mean for the before and after period. So in this case, they found the mean R ratio for the before period was, I think, 0.83, and the after period was 1.13. So to come up with their final, prediction of how much the treatment increases survival, they come up with a final ratio between the after and before period. In this case, it was 1.36. And that tells you that the mean survival in the treated reach increased by 36%. But this table is really, where the managers I work with have been kind of excited that we can get to this point. It shows the estimates of probability of juvenile survival and density increase or decrease a given percentage after BDAs are installed. So in this case, if you look at the column that says 50%, the probability that BDAs increase both survival and density of juvenile steelhead by 50% is 0.17 and 0.82 respectively. You can compare that to maybe the more moderate goal of increasing survival and density by 30%, which is 0.88 and 0.99. So there's a pretty good chance that you can increase both of those targets by 30% in this case. Okay, so for some lessons I've learned on how to do BACI successfully in this Northern New Mexico collaboration, but also a couple other projects I'm involved in, is collaboration is really important, including researchers at an early stage, to make sure that you have the best monitoring study design possible given the constraints of a specific project. So in my case, what this looked like was contacting managers from the regional office with the Forest Service and at national forest level and explaining that I really wanted to study the effectiveness of restoration and did they have any planned restoration treatments in the next couple of years that I could be involved in. But I think it can also go the other way. If you are someone that's going to be implementing treatments, reaching out to researchers with an agency or a university and asking if they're interested in studying the effectiveness of those treatments could be a good way to get a BACI study design in place. Communication is obviously really important. A clear plan outlining the goals, treatment types, the locations of the control and impact site, and a timeline that's shared to and agreed to by everyone involved is really important. So an example, from my work in Northern New Mexico, we have a really clear plan in place up in Valle Vidal. And so, it includes maps of where treatments are going and when they're going in that everyone has seen and is familiar with. And, apart from some delays due to COVID, that project has gone pretty seamlessly. But in comparison, the BDAs on the Santa Fe are being installed kind of as there is funding available and, volunteers or contractors available to do the work. And so the communication in that project hasn't been quite as strong. And we had a pretty, significant hiccup when some BDAs were installed in our control site. So communication can make a really big difference. That leads me to my last bullet point here, that flexibility and having flexibility and backup plans can help. So in that case, I had established multiple control and impact sites. So we were able to just kind of shift things around and we're still going to be able to use all of the data we collected. So it, wasn't totally catastrophic. But then kind of going into the plan with, ideas of how you might transition from like a BACI paired series approach where you're monitoring several times in the before and after period to maybe just monitoring once before and after, or being able to transition to if you lose your control site, can you transition to a long time series of before and after data? And then I think it's also a great time to think about potential ways to incorporate existing long-term data sets. One example in the Middle Rio Grande might be the Bosque Ecosystem Monitoring Program that has this long-term data set, and maybe it's been collected, at a potential impact and control site across time, bracketing when restoration has occurred. Okay, so now I'm going to take, a little bit of a hard right turn into the second topic that Katelyn suggested I talk about, which are potential metrics for evaluating ecosystem function. Restoration targets traditionally are often compositional or structural, and there's been a lot of studies that have shown that achieving these goals does not necessarily result in recovery of ecosystem function. In addition to that, metrics for evaluating function are rarely included in monitoring plans, and so the impacts of restoration on function are not especially well understood. So, when you are planning out your monitoring for a project, it's a good time to consider, if you could add in indicators of ecosystem function. Those might include, monitoring decomposition, primary productivity, respiration, nutrient uptake, or these last 3 bullet points that I have on my slide that I'm using in my work in northern New Mexico, functional traits and functional diversity, food web dynamics, and trophic transfers. So I'm going to go into those last 3 just a little bit more in depth before I wrap up. So functional traits are measurable characteristics of individual organisms that describe the response of a species to the environment and or its effect on ecosystem processes. You can use community level traits and functional diversity metrics to evaluate how restoration changes environmental filters. So that might, in a stream system, that might look like changes in depth to groundwater, or the effects of changes in the community on ecosystem function. So maybe that looks like, changing the abundance of nitrogen fixing plants. Functional trait databases already exist in a lot of situations, so you don't necessarily have to measure the functional traits yourself. I'm familiar with databases for plants, fish, and aquatic macroinvertebrates, but my guess is that there's more out there. And the benefit of this approach is that it relies on the community composition data that's already collected for many restoration projects. So you can take data that you probably already have, get into those databases, find the functional traits, and come up with some results looking at how restoration may have changed environmental filters or the community impact on ecosystem function. Secondly, are food webs. So restoration may increase the number of trophic pathways by reestablishing natural disturbance regimes, increasing habitat diversity, and altering community composition. And while recovery of food webs is really suggested quite often as a restoration target, it hasn't been applied in very many studies. This figure is taken from a paper by Locke that came out 2 years ago that found, I think it's 62 studies had looked at trophic structure, and of those, only 6 had used the BACI framework. So it's definitely an understudied area. In my work, I'm using stable isotope analysis to evaluate changes in trophic structure related to restoration. And then finally, trophic transfers, or flows of organisms, nutrients, energy that cross ecosystem boundaries. Subsidies like this can be highly variable across space and time, but they have significant impacts on ecosystem function and consumer resource dynamics in the recipient ecosystem. So the magnitude, quality, and extent of subsidies depends on several drivers that include boundary permeability, mobility of organisms, the size, shape, and similarity, distance between, and relative productivity of adjacent habitats. So all of these drivers are potentially impacted by degradation and restoration. So as an example in my work, I'm looking at aquatic to terrestrial subsidies. So, emerging aquatic insects that come out of the stream and then are consumed out in the riparian area, and how that changes with restoration. In my case, I'm using stable isotope analysis again, but there are also, other methods for studying consumer diets that may be possible for use in other restoration studies. So that's, my last slide, a quick presentation, but I'm really happy to take questions.
Well, thank you very much, Katie. That was excellent. I guess I'll start us off. And if anybody wants to jump in, you can either raise your hand, which I think I can see, or you can type your question into the chat. But I had a question going back to your first section on the BACI designs and the model, the different choices of models with your work with different managers. And so you indicated that your experience has been that at least recently managers are leaning more towards the Bayesian over the frequentist approach. And I was wondering, does that have something to do with the interpretability and applicability of the results? And if you could talk a little bit about how those different models treat uncertainty, and just why do you think it is that folks prefer the Bayesian approach?
Sure. Yeah. I mean, in my experience, I think it's exactly that. It's like the interpretability of that. So I think, you know, being able to, I think the Bayesian approach maybe better expresses, you know, how biologically relevant the change in the system is. And so, you know, if you come up with an effect size with the traditional approach of whatever, you increase stream temperature by 6 degrees, I think you're kind of left with this question of like, okay, how relevant is that for the biota in the system? Does that really matter? Whereas if you're setting a goal at the beginning of, you know, your restoration, that's like, you know, we really want to increase the survival of fish species by 30%. Then the Bayesian approach can really tell you like the probability that you actually achieved that. And so I think, you know, it's a way, it's more consistent with the way the managers, at least that I'm working with, think, you know, like we want to increase the cover of wetland plants by this much. And then, you know, you can express the probability that that happens versus you did, you know, increase the cover by this much. Is that, you know, meeting the goal that you set out at the beginning?
Yeah, and with that in mind, do you think it would be helpful? You also talked about communicating early about the approach and what the goals are going to be. So do you think it would be helpful before you even collect any data to sit down with the managers and say, okay, here's what this approach can give you. Here's what the results will look like. And then use that hypothetical as a way to set those thresholds and talk to the managers about their expectations?
Yeah, exactly. I think, you know, with my work in northern New Mexico, that's basically exactly how it played out is, you know, I approached the managers and said, I'm really interested in studying restoration. And if I can work with you to identify sites where we can use this study design, here's the end result that we can end up with. And, you know, they were pretty excited about that. And like I said before, especially, you know, the Bayesian output of like, oh, wait, we can express it this way, which really makes sense to, you know, managers and whatever audiences they're presenting their results to as well.
Yeah, that's fantastic. And then in terms of the metrics that you talked about, can you talk a little bit more about choosing metrics that actually respond to the treatment? Because you know, there's sort of these go-to metrics that folks use, or they, you know, you find in a lot of the literature. But I was wondering if you could talk about maybe your experience in choosing metrics that will particularly respond to your treatment, because not all fit all situations.
Yeah. Yeah, that's a good question. I think one is, you know, to start by thinking about what responds within the time frame that you have to do the monitoring. And, you know, there may be, you know, changes in the plant community may take a lot longer than something like changes in the macroinvertebrate community. And so figuring out what's appropriate for the time frame you have. But then, I guess I think that's probably the biggest limiting factor is kind of the response time. And then I think there's some indicators I think that can overlap between the structural and functional metrics. So I think, like I mentioned with the functional traits, that might be something that you're already collecting, you know, if you have population abundance data and community data, that you can do both without expanding your monitoring effort. I think that's something that food webs can also get at. You know, you're collecting population and structural data, and then if you can take that one step further and do some diet analysis, you can get to the point of analyzing food webs too.
Yeah, that's really what I liked about what you said about some of these indicators or these response metrics. They're sort of cumulative, so they cross over a wide swath of different effects, but through their sort of cumulative response. So you're not, I think it dampens down some of that natural variability in like say one species or one area, and it kind of gets that broader impact, which is kind of, I think, what we're all going for. on a lot of these restorations. So that was very helpful. Okay, we got a question from Tom Turner. He says, great presentation, Katie. Thanks. Is there a role for museum collections and specimens for documenting treatment effects?
Totally. I think, you know, kind of when I mentioned long-term data sets, I think there's a great opportunity to think about museum collections. And there's a good chance that you can, you know, kind of reverse engineer them into a Backy approach if they've been collected at different sites across time. And if you end up having collections that have been collected in a site that's been treated, as well as a site that hasn't, across time that, you know, brackets when restoration occurred. Yeah, I think there's totally an opportunity to use museum collections to evaluate restoration effects.
That's a great question and near to my heart, because so many museum collections are being moved around and discarded to make real estate for other things at universities. So I really appreciate that question. All right, any other questions? Feel free to unmute yourself and just we'll open it up to everybody. Oh, we have a question from Valley Girl. Do most land managers you've worked with have the staff and expertise to do Bayesian or more advanced types of monitoring, such as trophic?
I don't know. I guess I think several of the managers I know like don't have any problem understanding when I'm talking to about it. But yeah, I think the time is probably a larger constraint, especially at getting to the point of, you know, maybe understanding like R and all of the statistical programs to use those. But that's also why I think, you know, early collaborations with researchers, either at agencies or universities or grad students, is a really great way to make these type of approaches accessible. So, you know, in my case with the managers in Northern New Mexico, you know, they're not going to have to, I'm going to do all of that analysis and just send them the results. And they're in a situation where they get to use my monitoring to meet the monitoring requirements of their permit. But then they also get to, you know, share these results, you know, with their communities and everything to show how effective their treatments are. So I think that's a case where it's really important to have the collaborations in place and bring in all of the skills you need early on.
Yeah, that's a really great question logistically, and something to think about in the beginning. Okay, we have another question from Mick Porter. Has anyone compared a split plot analysis with the Backy design?
I'm not sure what a split plot is.
Yeah, that's one of the, if you look in some of the old experimental design books, that's one that's been around for a while. And so Mick, I'm not sure that Katie might have that experience, but I'm actually, I mean, you could probably find some comparisons between those. It would probably be particular to the situation, the setup of the design, but that's a good question. All right, anybody else have questions for Katie?
This is kind of a larger question. This is Yasmine, and please forgive me because I'm not a scientist by training. But, you know, how, you know, the work that you're doing in Valle Vidal on these smaller stream systems and wet meadows, you know, how could these approaches translate to a bigger river system like the Rio Grande? I think that's something that we struggle with because there's so much variation between different reaches and reasons for that, right?
Sure, yeah. You know, I think it really comes down to having like a solid experimental design. I think, you know, there are certainly challenges to working, you know, down in a larger system like the Rio Grande. Maybe, you know, you're more likely to have your plots disturbed by humans or whatever. But I think, you know, if you start with a really solid experimental design and come up with, you know, the parameters that work within your system, that you can definitely apply, you know, a similar approach to what I'm doing up in, you know, these like headwater systems.
Thanks.
So Katie, can you stop sharing your screen? And I think we'll be able to see folks. Here we go. Okay, did I miss anybody's question? Okay, I have one request for you, Katie. Could you maybe send me that Connor et al. Paper? I'd like to read that and see what the details are on that Bayesian approach. And then if everyone wants, I'll share it with the program. If that's possible. We have some copyright issues maybe, but I'll at least put the citation out for everybody to look up, because I think we might be interested in reading that paper. And the other thing is, can you type if you don't mind typing your email in the chat, so if anybody has questions after the fact, they can get in touch with you?
Yeah.
Thanks. All right, any more questions for Katie before we let her go? All right, thank you everyone for participating. We really appreciate it. And thanks, Katie, for your time.
Thank you for the invite.
Okay, we hope to see you again. All right, thanks so much, everybody.